All right, here's where we left off. We had just finished talking about what a hypothesis is, and we have three different examples here. If you saw the same phenomenon, this plant that I planted in my garden, it grows really well here, doesn't grow as well there, then you could come up with at least three different hypotheses. These are okay as hypotheses. You may want to phrase them in the form of an if-then statement. So for hypothesis one, you would say, if I grow plants in soil that is wetter, they will not grow as well as if I grow plants in soil that is drier. Or here it would be, if I look in the sunny areas, I will find more nutrient in the soil than if I look in the shady areas. Or in this one, if I grow plants with brighter light, then plants will grow better than in dimmer light. Okay, so there's our hypothesis. Now let's talk about experiments. The experiment is what we do to test the hypothesis. When we test a hypothesis, we're saying that's all well and good. You've got your suggestion for why the plants are growing better or not. Prove it to me. And when we say prove it to me, we're really saying perform a fair experiment so that when the experiment has been done, it only is logical that your hypothesis is correct. All right? So a couple of things about experiments that you need to know. You need to know about controls and about variables. Let's start off with controlled variables. All right. We'll get back to controls in a second. Okay? Variables. Variables are all of the things in this experiment that could be different. So let's talk about, okay, I'm going to do an experiment, and I'm going to do the experiment where I'm testing how bright the light is, right? I need to make sure that all of the plants that I'm testing, they're all as identical as they can be, right? It wouldn't be fair to have cactuses growing in bright sunlight and have ferns growing in dim sunlight, right? That wouldn't be fair. So there are some things, if you want to prove that how much light is changing how quickly the plants grow, then one of the things you need to do is keep everything except how bright the light is the same. And all those other things are the controlled variables. So you, as the experimenter, as the scientist, you need to make a special effort to keep everything else constant so that those things don't affect the outcome. So I'm telling you that I think brighter light makes plants grow better. So I need to start off with as genetically identical the plants can be, lots of them. I need to put them all at the same temperature. It's not fair to have some in warm conditions and some in cold conditions, right? I need to keep the temperature the same. They should all be in the same soil. It wouldn't be fair to have some soil that's really good and some soil that's really bad. That wouldn't be fair. We got to keep the soil the same. They should get the same amount of water, right? So in my experiment, if I want to prove that the brightness of the light is what makes the difference, I got to start out with all the same plants. That would be a controlled variable. They should all start out in the same soil. They should all get the same amount of water. They should all be at the same temperature, right? All of those things should be the same. Those are my controlled variables, okay? Now, what are the other variables? So the controlled variables, by the way, the controlled variables, you can have as many as you like, right? As a matter of fact, the better an experiment is, the more controlled variables you have, because that means the more things you're keeping identical so that someday in the future, someone's not going to say, oh, you say it's because of the brighter light, but actually the brighter light caused a warmer temperature. So it's really the temperature, right? Then you can go, no, I kept the temperature the same, right? So the other variables, one important variable is the independent variable. The independent variable is the thing that you are testing. It is the thing that you will be changing among the conditions. So in this situation with the bright light thing, the independent variable will be how bright is the light. So all of the controlled variables, the same plant, the same temperature, the same water, the same soil, that's all the controlled variables. And the only thing that you will change is the independent variable. You're going to give this much light to this group, twice as much light to this group, 4 times as much light to this group. Okay? That'll be your independent variable. Then the factor that you will measure, that's the data you will collect, that will be the dependent variable. And in our plant situation, we might be measuring how tall the plants got over 2 weeks and how many leaves they had. Okay, that would be my dependent variable. So in the dim light, plants grew 6 inches and had 12 leaves. In the twice as much light, plants grew 10 inches and had 20 leaves, right? That would be my dependent variable. The dependent variable is what you're measuring, okay? So the independent variable is what you're testing, right? If you decide it was how much water, that would be the independent variable for that experiment. If you decided it was how much light, that would be the independent variable for that experiment. Ideally, there should only be one, is that the next slide? One independent variable, okay? As many control variables as you like, only one independent variable. How many dependent variables? That can vary, okay? So a scientist changes one factor, the independent variable, and observes or measures what happens, which is the dependent variable. Okay, good. So I mentioned controls and controlled variables. Okay, that's confusing. I apologize. What are control groups? Scientific experiments often have a lot of control groups, but basically a control group is a group that gets chosen because we know what will happen to that group. If we know that in a certain situation plants won't grow, then that would be our negative control, right? So let's say I wanted to prove that water was making my plants grow better, then I might have plants that I don't give any water right from the beginning. That would be my negative control group. That would be take away all the water and I know things are going to die. Why would I do that? Why would I deliberately kill a plant? Well, in the plant situation, it's a little bit obvious what would happen. But in many of our scientific experiments, we're not entirely sure about any hidden variables, okay? So let's imagine that in my greenhouse where I'm not deliberately watering any of the plants, but let's imagine it's just super humid in the greenhouse. And so when it gets cool at night, there gets dew formation and that's kind of watering the plant. If I had a negative control group where I never gave any water through the whole experiment and those plants didn't die, I would say, ooh, my negative control didn't work, all right? Now, these negative and positive controls, they're not being tested, but they are being measured. What do I mean by they're not being tested? Not being tested because we know what to expect from them. They are being measured so that we can say, if I give water to this plant, it grows twice as fast as no water or something like that, okay? They're used for comparison. In lab experiments also, we very often, and you'll be doing this in your lab this semester, that when sugar is present, the test will turn a different color, okay? So one of my positive controls would be a test tube full of sugar water. If I use a test tube full of sugar water as my positive control, and I do the experiment and it doesn't turn the right color, I would go, uh-oh, we've got the wrong chemical here, okay? So positive and negative controls, just make sure that there's not some hidden mistake in what's going on. So examples of variables. If you want to test the hypothesis that more sunlight makes plants grow faster, then you would grow identical plants in exactly the same soil with exactly the same amount of water. Those would be 3 of our controlled variables, but with different controlled but different amounts of light each day, one independent variable. And what would you measure would be the dependent variable, and we might say the total weight of each plant after 3 months of growth, right? Our control. For a positive control, you could grow an identical plant in that spot in your garden where you know that they grow really well. And for the negative control, you could grow an identical plant in exactly the same soil with exactly the same amount of water and stuff like that, but with no light, because you're sure that won't grow. See, negative control, you're sure growth won't happen. Positive control, you're sure that really good growth will happen. And then your experiment will be compared to those. Okay? So, also, scientists always repeat experiments many times, and they even have asked scientists at other universities to repeat the experiments so that there are different groups of scientists that are trying the same thing in different hands. And we do that before we feel we can analyze it and publish it. For example, for those of you who have read my scientific papers, oh, I know you have. No, of course you haven't. But in my scientific paper, I published that this protein that I discovered would cause bacteria to die. And a different group of scientists using a different source of that protein, they did those experiments and they said, you know, what we found is that it'll keep bacteria from growing, but it doesn't actually kill them. And I was like, no, I'll repeat it again. And it did kill them. He repeated it again. It didn't kill them. So we got our heads together and we were like, why aren't we getting the same results? And it turned out that my protein I got from a natural source. I got actually from a living creature. And in their experiment, they were creating it in a laboratory. They were building the protein by a cloning technique. And they found out that they were missing one ingredient in their protein. And if they added that ingredient, then it's true, the protein did kill bacteria. And so they ended up validating my experiment, right? So that's what we always want to do. What's the difference between data and results? Okay, data are the numbers from your experiment. This amount of light on average to plants grew 6 inches. This amount of light on average to plants grew 10 inches. That's the data. What are the results? They're the conclusions, right? That the plants, as they got more sunlight, they grew faster up until they got this many watts. And after that, no matter how much extra light I put in, they didn't grow any faster over the 3 months. Okay, those would be my conclusions. So the data, the numbers, or whatever you're acquiring, the results, that's your conclusion. What does it prove? Did you need to reject your hypothesis or did you accept your hypothesis? What if the data of your experiment are not what you predicted? Okay, it does not mean that you're a bad scientist. One of the first things that my first mentor taught me in science is don't argue with the data. Your data are your data. It's okay to go ahead and repeat an experiment, make sure you didn't make any mistakes. But once your data is there and it says no, for example, more light does not make the plants grow faster, then you need to reevaluate your hypothesis, okay? So you don't argue with the data. If it contradicts your hypothesis, repeat it once to make sure you did it correctly, but then you need to change your hypothesis. Technically, if it contradicts your hypothesis, you need to reject your hypothesis, and then you say, well, what next? What's your next hypothesis? For example, it could be, we had 3 hypotheses for why the plants might have been growing faster in the sunlit part of the garden, right? We thought maybe more nutrients in the soil, maybe more sunlight, maybe more water. If I test the sunlight and it doesn't make them grow faster, We'd go to one of the other hypotheses, we would test that. And by the way, if you're a scientist and your data does not your support hypothesis, you may have actually discovered something really interesting. Another thing that my mentor told me really early is that great discoveries don't sound like, I knew it. Great discoveries sound like, well, that's weird, right? So, as a review, scientists are constantly solving problems, and they do it with the scientific method. Okay? You identify the problem, make an observation, then you form a hypothesis, very often an if-then statement, design an experiment to test the hypothesis, collect the data, decide what it means, what are your results, what is your conclusion, retest it, and don't forget step 8. Is it on there? Don't forget step 8. It will be on there very soon. Step 8 is communicate your discovery. All righty, so now you have got other stuff to do in your lab other than watch this lecture. Get to all of that. If you've got any questions, make sure you come to an office hour on Zoom and I will walk you through how to submit your data or whatever it is you need help with. All right?